Hello students, today we'll study the chapter Microorganisms, and in this chapter we will study protozoa. So here some general characteristics of protozoa. First thing is the protozoa are found in the aquatic environment, right? So protozoa, just a minute, please let me take the pointer first. Okay. So protozoa that are found in which environment? That is aquatic environment. That is aquatic environment. And they live in fresh water or oceans, and some are free living and some are parasitic in plants and animals. And mostly they are aerobic, but some are anaerobic and present in the human intestine. So here this point is very important, human intestine. Now, some of the species are found in extreme environments like hot springs. Some of them form resting cysts to overcome dry environments. Okay, so protozoa here. Now, protozoa, first thing it is found. Protozoa, it is found in the aquatic environment. Protozoa that is found in the environment. Which environment? That is in aquatic environment. So here I am writing here. So the protozoa, those are actually microscopic, unicellular, eukaryotes that have a relatively complex internal structure. Okay, it is not so much small and unicellular, but eukaryotes, and it is complex structure, and internal structure is a little bit complex, and it carry the complex metabolic activities, and it carries complex metabolic activities, complex metabolic activities. And some protozoa have the structure for propulsion or other types of movement, right? So protozoa, these are microscopic unicellular eukaryotes, and it has relatively complex internal structure and rather than other microscopic unicellular eukaryotes, and it carries the complex metabolic activities. Here in case of protozoa, it is found in aquatic environment. It is found in aquatic environment. And on the basis of light and electron microscopic morphology, the protozoa are currently classified in 6 phyla. Okay, so we'll study those things later on. So for the first thing that we are knowing that the protozoa that are found in the aquatic environment, and they live in the fresh water of the oceans. They are free living and some are parasites, and that means parasitic, and this nature they have, and mostly they are aerobic, but some are anaerobic also, and it present in our which organ? That is human intestine. That organ, that is the human intestine. And some species also we can find out in the extreme environments like hot springs also. Can you imagine that thing? So first thing, what is the protozoa? Like whatever we have studied till now, some fungi, and also we know name of so many microorganisms. This is also one microorganism. So this protozoa is the microscopic unicellular eukaryotes, and it carries the complex metabolic activities. And also this protozoa, it has relatively little bit complex internal structure. Okay. Now the size and shape. The size and shape of the protozoa vary greatly, and from microbial 1 micrometer to large enough. And here 1 micrometer, this is nothing but 10 to the power minus 6 meter. Okay, this is the shape that is the from 1 micrometer to large enough, and we can see it in the naked eye, but most of the cases we have to see it with the help of the microscope. So we can find out what are the portions of the protozoa. Now, the shell of unicellular foraminifera can have a diameter of 20 centimeter, and they lack a rigid cell wall, so they are flexible and found in various shapes. So they are in various shapes because these protozoa, these have some lack of rigid. The cell wall is not so much rigid, so it can change its shape, and cells are enclosed in a thin plasma membrane, and these cells will be enclosed in a thin plasma membrane. So protozoa, these are one-celled animals and found worldwide in most inhabitants, and most species are free living, but all higher animals are infected with one or more species of the protozoa. And infections, do you know that thing? The infections range from asymptotic to life threatening depending on that species, this type of species, life threatening disease. So it can cause due to this thing, this protozoa. So protozoa that are considered to be the sub kingdom of the kingdom Protista, and all whatever the Protista, that the protozoa are considered to be a sub kingdom of Protista. Okay. Next, here you can see the structure of the protozoa. Here, some of the species have a hard shell on the outer space, but generally the cell, whatever the outer surface of the cell, that is not so much rigid. That's why different sizes we can get. And in some protozoas, especially in ciliates, the cell is supported by pellicle. The cell is supported by pellicle. Here you have the pellicle. You can see this is the support, and which is more flexible or rigid. It can be more flexible or rigid, and it gives the organism a definite shape and help in locomotion. And it helps in locomotion, right? So this pellicle, this actually helps for the locomotion of the protozoa. Okay, so this is very important thing. And now next thing is that the cellular structure. The cellular structure here, they are unicellular and having eukaryotic cell. Already I have told you that they are unicellular, that means single cell. And eukaryotic cell, already I have told you, if this is membrane surrounded, at that time eukaryotic cell. And the metabolic functions also I have told, the metabolic functions are performed by some specialized internal structures, and the metabolic activity is very complex here, and these mostly have one membrane bound nucleus. Mostly it has what? One membrane bound nucleus in the cell. So here you can see the protozoa, the pellicle, which helps to give one shape, and here you can see the cell membrane. This is the cell membrane, and different sizes also, it can be of different sizes due to this cell membrane. It is not rigid, so at that time it can take the shape of different types. Okay, so it can take the shape of different types. Now the nucleus, whatever here, this is the nucleus, this one. Now this nucleus, this one, it has diffused appearance due to scattered chromatin. Due to the what? Scattered chromatin. And the vascular nucleus contains a central body called the nuclei or endosome, or it is called endosome, and nucleoli is epicomplexons have DNA, whereas amoeboids lack DNA in their endosome. Okay, it lack their DNA in the endosome. So here in this picture you can see that this is the nucleus, and this nucleus a little bit diffused appearance it has due to the scattered chromatin. And in the life cycle, I can say that the stages of the parasitic protozoa actively feed and multiply are frequently called the trophozoites. And in some protozoa, other terms are also used for these stages. That cysts. Cysts are stages with a protective membrane or thickened wall. Cyst, what is cyst? That cyst is cysts that are the stages with a protective membrane or thickened wall. And protozoan cysts that must survive outside the host usually have more resistant walls than the cysts that form in tissues. Okay. So ciliates, already we know the ciliates, and ciliates, they have the micronucleus and macronucleus. The plasma membrane that encloses the cytoplasm and other locomotory projections like flagella, pseudopodia, cilia. Here you can see this thing in this picture. You can see this thing. Here you have different, this is the nucleus, and here you can see different parts also. I will show you in the picture. And in the ciliates we have the micronucleus nucleus and also macronucleus, the plasma membrane that encloses the cytoplasm, and locomotory projections like flagella, pseudopodia and cilia. And some of the genera have a membranous envelope called the pellicle. Pellicle already I have told you, and it gives definite shape to the cell. Here you have the cell, and this is the cell, and there is a nucleus here, and this is the nucleus here, and this is the actually pellicle. This is the actually pellicle, the cell wall. In some protozoa, epibiotic bacteria attach to the pellicle by their fimbriae. Okay, so in some protozoas, epibiotic bacteria attach to their pellicle by their fimbriae. And this cytoplasm is differentiated into outer ectoplasm and inner endoplasm. So here in the protozoa, in case of protozoa, you can see that there will be, in this case, cytoplasm. It is differentiate the cytoplasm. It will be differentiate here. You can see, suppose this one is the protozoa. Now I'm not drawing all those things. So here, let me take another ink. I will show you. Let's take green. Okay. So here, this is the macronucleus. Okay. And this is the macronucleus. This one is the macronucleus. And we have, let me take another color. And we have, let it be, these are micronucleus. Just a minute, these are actually filled. Just a minute. Okay, so here these are micronucleus, and I'm just drawing one rough one. And here you have these are cytoplasm. These are cytoplasm. And there will be a lot of, just a minute, let me change the ink color. Here you have Okay, all over this. I'm not drawing the whole thing. The main parts I'm just showing here. The main parts I'm just showing here. So micronucleus, you understood what is micronucleus and macronucleus also. And these are cilia. And this one is actually this cell wall. This is actually pellicle. Okay, this is actually pellicle. And another thing I can say that there are some food vacuole also, and anal pore also it has. So generally these are the main things we have. The cytoplasm, that is differentiated in the outer ectoplasm and inner ectoplasm. And ectoplasm is transparent and endoplasm contains cell organ, and some of the protozoa have cytosome for ingesting foods. It has also cytosome for this. Here you can see that cytosome, the cytostome. Cytostome, here you can see here. I can draw here. Let it be this protozoa. Now here you can see there is a part. This is the part. This is the part, and this part. Just a minute. This part. This part. This yellow part. This part is called cell mouth. Cell mouth or cytostome or cytostome. Okay, so to ingest the food, right? And food vacuoles also present. So I'm showing you the food vacuoles. This one is one food vacuoles. Another food vacuoles you have here also. Another food vacuoles. So these two are food vacuoles. Food vacuoles. So these two are food vacuoles. Here you can see the food vacuoles are present where ingesting food comes. So here this is the cell mouth through which this is also called cytostome, and through which the protozoa, it is just taking the food and ingesting food, and then this food ingested that food will come to the food vacuoles. Okay, so these are the things. Next, here it has been shown macronucleus. This is the cell mouth, and this is the cell mouth, and this is the buccal overture. Like we have the buccal cavity, this type of they have also mouth, and this they have the buccal overture, and this is the cell mouth to ingest the food. This is the cell mouth to ingest the food. Let me take another color here. And here you can see the cytostome, and here food vacuoles you have, and the ciliates have the gullet and a body cavity which opens outside, and the central vacuole is present for osp regulation, that removes the excess water. This central vacuole that will just removes all the excess water. And membrane bound cell organelles like mitochondria and Golgi bodies, lysosomes and other special structures are also present. Mitochondria and also other bodies also are present. Mitochondria, Golgi bodies, lysosomes, these are also present. Okay, so here there are different types of protozoas that we have shown. Next is the nutrition, how they are taking the nutrition. Now, protozoa are heterotrophic and have the holozoic nutrition. These are the heterotrophic and holozoic and what holozoic nutrition. So, here you can see that this protozoa, in the right hand side you can see that this is a food particle, this is a food particle, this is the food particle, and here you have the entrapment and entrapment, and this is the cytoplasm, and this one is the nucleus, this is the plasma membrane. So it will take this thing, and then whatever it will do, it will just grab this food, and this it will just grab this food, then it will create one, due to the fusion of with the lysosomes, it will create one wall, and then it will digest. Okay, so these protozoa, these protozoas, these are heterotrophic and have the holozoic nutrition. They ingest the food by phagocytosis, and this is the phagocytosis cycle. Now, some of the protozoa group have a special structure called cytostome for phagocytosis. What? That is the mouth actually. That is the mouth of the protozoa. And the pseudopodia of the amoeboids help in catching the prey. And here you can see that this type of structure, this will help to grab the food. Okay. Now, what happens? Here, thousands of cilia that are present in ciliates drive the food-laden water into the gullet, and ingested food comes to the food vacuole, and it will get acted on by lysosome enzymes, and the digested food gets distributed throughout the cell. Okay, so here you can see that whatever in the phagocytosis, what happens? If you have the protozoa, that is heterotrophic and holozoic nutrition. So main thing is that the mouth they are using to take the food, right? That is the that is our cytostome. Okay. Now this phagocytosis, this is the process through which you can see that a cell uses its plasma membrane to engulf a large particle. This is the large particle, right? So here, this phagocytosis, in this process in which a cell uses its plasma membrane and to engulf the large particle, and then giving rise to an internal compartment called the phagosome. Let me, it is a little bit enough. So here you can see that I can write this thing. Here first thing is the phagocytosis. This is the process. This is the process by which what happened? Cell uses their its plasma membrane. It is eukaryotic, right? So it has the plasma membrane, and it uses to engulf the food, large food. So a large particle of the food, large particle, right? And then what happens here? The internal component and giving rise to an internal compartment, and it giving rise to an internal compartment called phagosome. Okay, and it is one type of endocytosis. So a cell performs the phagocytosis is called phagocytes. A cell which performs this phagocytosis, that is called the phagocyte. Okay, So phagocytosis, that means the cell membrane, it will use the cell membrane to engulf a large particle. Then what it will do? It will just create one internal compartment that is called the phagosome. That's why this phagocytosis, this thing it comes. This thing it comes. Now the phagocytosis, that is, this is the process, and how the we have seen in the picture also, in the image also, how it is taking the food and large particle it is taking, and then this large particle it will just I'm trying to draw here. See here. So here, this is the plasma membrane, okay? This is the plasma membrane. And here we have a food. This is the food, large particle of the food. Now what it will do? Now, oh sorry, just a minute. This is the vacuole. Okay. Now you have the food here. Whatever the food it has taken, now you have this food here. Now you have the food here. Okay. Now it is clear how they are taking the food using the plasma membrane. Now the locomotion. So most of the protozoa, these species having the flagella and cilia or pseudopodia. Okay. So here the locomotion, if we see, that means it has this. It has some flagella or cilia. Cilia already we have seen. Now, these sporozoa, which don't have any locomotory structure and having subpellicular microtubules, which helps for the slow movement. Here you can see how they are. These are the locomotory organelles and locomotion methods. Here you can see how they are just moving, how they are just doing their locomotion. So here, if we see the picture, locomotion of the protozoa in this case, this exhibit the diverse mode of the locomotions across various groups. And now here you have flagellars, ciliary and amoeboid movement. Here you have, I am writing here, flagellar, ciliary movement also there, and amoeboid movement. Okay. So only the ciliates among the three major these movements, only it major group of protozoas they are doing this thing, and these are monophyletic group. Okay. So here in this case the protozoa, they have three types of movement: flagellar, ciliary and amoeboid movement. And here flagellar propulsion, in the flagellar propulsion it is employed during some stages in the life cycles of the certain amoeba. Okay, some amoeba that they are following this thing. Now for the cilium structure already we have seen. Now amoeboid movement also we know, this type of in depth we do not have in the syllabus. So you just remember these three types of the movements it is possible in the protozoa, and these are the pictures also. Now the life cycle of the protozoa. The life cycle of the most protozoa alternates between dormant stage to the proliferating vegetative stage, or example, that is the trophozoites. Okay, so it will just alternates between dormancies to the proliferating vegetative stage. And the cyst stage can survive harsh condition without water and nutrients, and it can remain outside the host for a longer duration and get transmitted. The trophozoite stage is infectious, and they feed and multiply during this stage. This trophozoite stage, this is very dangerous stage, and this is very infectious stage. And here they will just take the food, and here they will multiply themselves. So many protozoas are there, and here you can see that due to this stage, we are suffering. We are suffering very much for this stage. So here in the life cycle, we have seen that we have in the life cycle of the protozoa, we have that two that will alternate between two stages. One is the dormant cyst stage, another is the trophozoite stage. And during this its life cycle, a protozoa generally passes through several stages that differ in structure and activity. Trophozoite, this is the Greek letter. This one is the Greek letter. So in the life cycle of protozoa, that we have it will alternate between two stages. One is the dormant stage and another is the trophozoite state. Now let me tell you one thing. So let it be first, let it be this is early cyst wall formation. Okay, this is early cyst wall formation. Okay, then this is, here you can see, just a minute. Here you can see this one is the mature cyst. Mature cyst and dormant and in the resting stage. This is dormant and the resting stage. Now whenever it will get moisture and nutrients restored and nutrients, then cyst wall that will break. Here you can see that cyst wall this cyst wall that will break. The cyst wall that will break. Okay. Then so here the cyst wall break, open, and then the trophozoite is reactivated Then it becomes like this. There is some granules here, and here so after this, so here you can see, just a minute, let me draw this. Now this is drying and lack of nutrients. Okay, lack of nutrients, and this is the trophozoite and active feeding stage. This is the trophozoic and active feeding stage. Trophozoite and active feeding stage. So this is the total life cycle of the protozoa. Okay, so here you can see that here cell rounds up and loses the motility. Here the cells rounds up and loses the motility due to the drying and lack of nutrients. Then what happens, this mature, this cyst are dormant cyst. Now whenever it will take the moisture and nutrients, restored nutrients restored, then cyst will break, so and it will open, and then trophozoite is reactivated. And here you can see this is the stage where trophozoite is active and feeding stage. Okay, and this is the feeding stage. So this is the total life cycle of the protozoa. Okay, so the cyst stage that can survive harsh conditions without water and nutrients, but it is dormant state. But in case of trophozoite stage, we require moisture as well as the nutrients. And here in this condition, the trophozoite stage, that is very infectious, because here it will take the food and it will multiply during this stage. During this stage it will multiply. And now the reproduction. So this reproduction process we will study in the next lecture. Till then, it's over now. Thank you and bye bye.